Hi there, and welcome to today's video. Last time we discussed what synthetic biology is, how it can make the world we live in a better place, and we introduced you to some old school methods like classical transformation and transfection with plasmids. Today we'll have a look at more recent methods in the world of genome editing. One of the most well-known new methods is the CRISPR Cast technology, which is short for clustered, regularly interspaced short palindromic repeats. It is a gene editing mechanism that has recently revolutionized genetic engineering with its accessibility, ease of use, and fast rate. In the present time, scientists are already utilizing CRISPR to, for example, insert malaria-blocking genes into mosquitoes or in wheat to induce resistance to specific diseases. A possibility for CRISPR lies in eliminating genetic diseases, a process that is still being developed. Now, where does CRISPR come from? Unlike many inventions, CRISPR is not invented from scratch by scientists, but instead we learned it from bacteria, which use the CRISPR system against invading viruses. When a virus attacks a bacterium, it inserts its own DNA with a set of instructions into a bacterial cell. This way, these viruses hijack the cell and instruct it to copy the virus inside. These copies then burst out of the cell on their way to hijack other bacteria. As a defense mechanism against these viruses, prokaryotes have evolved a CRISPR-Cas adaptive immune system. In this system, the Cas part is an enzyme that functions as molecular scissors that disable the virus by cutting the foreign inserted virus DNA into two. Then the bacterium inserts a section of the viral DNA into a special area of its own genome that can be seen as a library of mugshots of hijackers that the cell uses to recognize the virus in the future in case it strikes again. If it does, the bacterium recognizes the virus and produces a personalized CRISPR system based on the mugshot to cut the hijackers into pieces. This way, the bacterium fights the viral DNA by breaking it apart. The scientists were inspired by these mechanisms and used it to develop the famous now CRISPR-Cas9 system. In the lab, a small piece of RNA is created with a guide sequence, which binds to a target sequence of DNA in a gene and to the Cas9 enzyme. The modified RNA is utilized to recognize a specific sequence in the DNA, and the Cas9 cuts the DNA at the right location. If the DNA is cut, scientists can use the cell's own DNA repair mechanisms to either add or delete genes, or change it by replacing it with a customized DNA sequence. Thinking about the applications of this genome editing technique, it can be a successful way of combating the upcoming resistance against antibiotics. In a recent animal study, researchers engineered a plasmid to remove an antibiotic resistance gene from a bacterium from the intestinal flora by using the CRISPR-Cas9 system. In mouse models, it reduced the abundance of resistance genes in threefold. Back in the day, genetic engineering used to be a non-specific process, but thanks to genome editing, it is now possible to work very specifically, which enables scientists to just knock out genes in a way that simulates natural mutations. Although people first think of CRISPR in genome editing, there are multiple alternatives that also have their own strengths, as CRISPR is limited by some factors. One viable alternative are other types of gene editing nucleases, such as, for instance, zinc finger nucleases that are based on zinc finger proteins. This is a family of naturally occurring transcription factors that are fused onto a nuclease. Two discrete zinc finger nucleases recognize and bind specific sites of opposite DNA strand. Then the assembled nuclease cleaves the target DNA and the genome can be edited. Because it has been found that zinc finger nucleases do not always bind very well, another type of protein was found that would resolve this problem. These proteins, known as Tall effectors, come from bacteria that infect plants. When attached to nucleases, They are called tollins, which is short for transcription activator-like effector nucleases. When one binding subunit contacts one nucleotide and the other binds to the complementary one, the nucleases cut the DNA. The advantage here is that the subunits do not interfere like in zinc finger nucleases. These engineered nucleases are very promising, but improvements should be made as it is not always possible to get the exact desired change. In fact, last February, a US trial has safely shown CRISPR gene editing on 3 cancer patients. So who knows? In the far future, we could possibly live in a world without diseases. Thank you for watching. Subscribe and stay tuned for more videos on real-life applications of these amazing tools.